Hi everyone, my name is Shania Prasad and I am the Scientific Sales Specialist at AD Instruments. Today our webinar will be about linking physiology concepts for biomedical engineering at University of New South Wales. Our guest speakers for today is Associate Professor Richard Vickery and Associate Professor Mohit Shivadasani. Associate Professor Richard Vickery is a researcher and educator in the School of Biomedical Sciences at UNSW Sydney, and he has been recognised with the Vice Chancellor's Award for Teaching Excellence. He has fostered a collaborative culture of innovation and commitment to continual improvement in education, while also focusing on understanding the internal language of the brain for his research. Associate Professor Mohit Shivadasani's expertise lies in bionics, especially devices that use electric stimulation to treat various sensory and neurological diseases. He was also a key member of the interdisciplinary team responsible for the design and preclinical testing of Australia's first prototype bionic eye, currently leading several research programs that aim to develop new devices for vision loss, chronic pain, and inflammatory bowel disease. Shivadasani also coordinates and teaches a course in bionics and neuromodulation that is taken by postgraduate engineering students. Before I hand it over to Richard, I'm just going to discuss a little bit more about our company. AD Instruments is based in Dunedin, New Zealand, and has been around for 30 years and specialized in hardware and software for data acquisition in research and education. Some of our hardware and software include the Education Power Lab 26T, which has a built-in fully isolated 2-channel bioamplifier, isolated stimulator, separate non-isolated analog input, and a differential mode analog output. This can be paired with our LabChat 8, which allows you to customize your workflow, acquiring biological signals from multiple sources simultaneously and applying advanced calculations. Some of our hardware include the human NIBP nanosystem, which measures continuous non-invasive blood pressure, our Trigno wireless foundation system, which can measure EMG and XYZ axis and a range of other different things with different sensors, our EcoVital wireless physiological system, which measure ECG, skin temperature, GSR, accelerometer, breathing trace, and SpO2, and our wireless heart rate measurement, which can be used alone or in conjunction with other equipment to synchronize with recordings such as ECG, HRV, and spirometry. Now I'll be passing it over to Richard.
All right, hopefully you can see that now. Does that look correct?
Yes, we can see it.
Yeah, look, apologies, everyone. Look, welcome. So I'm a physiologist, so I do research with Mohit, and we supervise students together. And in fact, we teach the same students in that we teach biomedical engineers, but I'm approaching it as a physiologist where they come and take my courses, whereas Mohit will be talking to you more as a biomedical engineer. And particularly, I am a neuroscientist, so my stuff is really going to relate to the brain. So let's get into it. So we're running a little late. So the teaching context here is that it's a large university, UNSW, so 65,000 students. We're research intensive. We have high quality students. We're producing high quality research, and we teach across a broad range of subject areas. I'm in the Faculty of Medicine in the School of Biomedical Sciences, and we teach science students, medicine students, allied health, and biomedical engineers. And we have a strong focus on hands-on experiential learning, which we do through practical classes. And normally they run 2 or 3 hours. That's one of the changes in modern education. When I was an undergrad, prac classes sometimes ran 6 hours, but that never happens nowadays. And our practical classes are often very large, so up to 115 students. And some of our cohorts, even with that size class, we have to repeat it multiple times. So this is one of our prac classes. You can see here we're doing a lab, and I'll talk about this one in a minute. But what we try and do is to get students to understand that the knowledge that we have is limited by the tools that we have to acquire it. So to understand how the body works, or in my case, how the brain works, we're limited by the tools that we have. And I'll take you through some of the pracs we do with students to show those sorts of limitations and the ways that we actually can acquire data. So we'll talk about large-scale recordings. I'll talk about some multi-neuron level recordings, some single neurons, and then we put all of that together in some labs, and I'll give a brief flavor of that. So large-scale recordings, here, example I'll show is EEG, so the electroencephalogram. There are caps available. We just have isolated electrodes on two hemispheres, and we get very nice signals out of these. So these are the sort of EEG signals that you can get. This is a student in a relaxed state. So this is what we would call alpha waves. So on both hemispheres, so we've got the left and the right hemisphere, we have this regular activity. And the students can count this. So this is 1 second. And for alpha waves, there should be a frequency of 10 to 12 hertz. We're collecting this using chart software, and that has some nice tools built into it. And so one of them is that you can do a fast Fourier transform. So here is the energy in that signal that we're requiring. And so you can see the energy has a strong peak here. This is hertz. So at 10 hertz, most of the energy is there, which is consistent with what we were seeing here with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 peaks. And then this is showing that frequency here as a heat map. So we can see there's a heat map here at around 10 hertz. There was some lower frequency activity here. And then this is showing you in real time. So you can actually see that scrolling and updating while the students are relaxed. And then as they open their eyes, that frequency range will spread over a much bigger area. So there's lots of things we do with EEG, but what we're particularly trying to do in this lab is to do some cortical localization. We've got the left and the right hemisphere. And so the relaxed activity, those alpha waves, are synchronized between the hemispheres. But what we do then is present flashing gratings. And if you present it to one side of the visual field, so if we present it on the right half of the visual field, then that will activate the left visual cortex. If we present it on the left, it will activate the right. And so then again, we do a Fourier transform looking for energy at the rate at which we're flashing that grating and see if we can see differences between these two hemispheres to prove to students that, in fact, the right half of the world is being processed here in your left occipital cortex. At a slightly finer scale, so instead of hundreds and thousands or millions of neurons, in EMG, we were recording from hundreds of neurons. So EMG is the electromyogram. You can do frequency analysis on that using the same tools that I just showed you, that FFT. But we do one quite nice lab, which I think is interesting to explore, which is where we look at motor control. And particularly in motor control, there are feedback controls. So if I'm reaching out to touch my finger, I can guide that by vision and by proprioception. But some things, like if you want to hit a cricket ball, you need to pre-program that. It's actually got feed-forward control. And so what we do, we have the students holding a button, which is going into one of the channels of the power lab. And so they're pressing the button against the bench, and then they're trying to make a movement as fast as they can. And that releases the button, which provides a trigger that we can see. So because they're doing that movement with a biceps contraction, we get a big biceps contraction as the button is being released. What we want to do, though, is that the student moves towards a target. So they move towards someone's hand and have to stop abruptly. And so to stop that movement, they need activity in the triceps muscle. So we're recording EMG from biceps and triceps. But then, of course, if you have a big triceps activity, you would have a recoil. So there's then some smaller biceps activity to null out the triceps, and then there's some smaller triceps to null out the biceps. And you have this alternating activity to let you do a fast motion. That in itself doesn't prove whether it's feed forward or feed backwards. So what we do is that we have the students do this movement like I showed you. They're moving rapidly, stopping at a target. Here's the biceps activity. As it releases, here's the triceps. Then after the triceps, we'll get some biceps. Then we get some triceps, then some biceps. What we do, though, then is that we trick the students. We have a strap around the arm which arrests their progress. So they don't need to do it. Someone else is holding this strap. But sometimes the strap will be loose and they need to control their movements. Other times the strap will stop them dead. And so when they're wearing the strap, we have exactly the same pattern of activity. So here's the biceps. We still have the reflex triceps to slow it down, even though they don't need it. The strap is stopping them. Still have this reflex biceps activity, which shows that we've pre-programmed 30 or 40 milliseconds of motion because we can't slow the muscle down quickly enough. So, and this is generated by the hundreds of hundreds of motor neurons controlling your biceps and triceps. Again, at that multi-neuron scale, we record what are called sensory nerve action potentials. So we can record at the wrist and the elbow here, and we actually do this over the median and the ulnar nerve. And this is a great practice we do with biomed engineers and with doctors, because we can diagnose things like carpal tunnel. We can measure peripheral conduction velocities. And so it's quite a straightforward setup. We're just using channels 3 and 4 on the power lab here, and we do some stimulation to find where the nerve is located to work out where to put these electrodes. We get some very nice signals from that. So this top channel is what we're recording at the wrist. And so this is a compound action potential. We call it a snap because when we're stimulating down at the digital nerve level, there's no motor axons in it. If we were stimulating higher up, it would be a mixed nerve action potential. And then here is that same action potential, but now recorded at the elbow. So it's been induced. Here's the stimulus artifact. This is the time for it to travel up the finger, reach the wrist, and then this is the time to travel from the wrist to the elbow. And so we can use this to get a very accurate measurement of the conduction velocity in different segments, particularly from the wrist to the elbow, because we can measure the time here, but we can also measure the finger to wrist time and look at changes and look at the effective temperature and so on. Then zooming into an even smaller scale, we do single neuron recordings. And some of these we've integrated into sort of electronic lab books. So this is a student notebook, and all of their pracs for the course are here. Some of these involve Power Labs, some don't, and students can click through to do the different subtypes of experiments. But what we're doing here is actually recording from single sensory neurons in cockroaches. And let me show you the setup. So here, we need a microscope to see it. We've got a stimulator that I'll show you in a little bit more detail, and we've got a cockroach leg that we have got some pins in and that are going to give us our signals. So this is a cork with the cockroach leg. Here are the recording pins going into the bioamp on the Power Lab. And what we have is just a simple speaker, and we're controlling this with the stimulus output of the Power Lab, so we can do controlled stimulation. So here's the cockroach leg. That's one of the pins. These are the spines on the cockroach leg, and we can activate just one spine in a very controlled manner by driving that little speaker through the output of the Power Lab. So what sort of signals do we get? Well, we get recognizable individual neurons. So the scale here is microvolts. So a good size signal is sort of 50 to 100 microvolts. These are from the same nerve, the same cockroach, just pushing on different spines. And you can see there's actually lots of different neural signals here. So there are some smaller ones, some middle-sized ones, but these ones we get the students to study in the class. So these are recognizably the same unit, and this is recognizably a different unit. So even though it's about the same size as this one, you can see it's got a double peak positively. It's got a very narrow negative phase. And so that profile of these individual neurons is really just down to their juxtaposition to the two recording pins. But it then lets us test whether this neuron responds differently than that neuron to a variety of stimuli, how they encode intensity, how they adapt to the prolonged stimulation, that sort of thing. We can also use this controlled stimulation to do conduction velocity measurements. So here we have the stimulator pulse that's driving the stimulator to touch the spine, and then here we've got a number of action potentials recorded from that nerve. This is in milliseconds along here. So this looks like, in this case, the conduction velocity is pretty much onset is at about 4 milliseconds. We measure the distance and we can work out the conduction velocity for that cockroach leg in much the same way that we do for the humans, but now for individual neurons rather than for a compound action potential. We use the same prep, which is cockroaches, but we can record motor activity. So they have these ganglia, which are groups of neurons. The thoracic ones are related to legs and to wing activity. And using the same technique with pins penetrating those ganglia, we can record very nice signals. So here we've got two different neurons, one bigger one smaller one. They're both firing away. And we test, in this case, we do a little bit of pharmacology as well. We put some drugs on to excite or inhibit those neurons, as well as looking at the interaction between them as they're firing away in these regular patterns. And then we use those tools to try and collect a variety of signals to do sort of engaging pracs where students have to really think hard about what sort of signals are happening. So here you can see a student, she's got a whole variety of things hooked up to her. She's got a finger plethysmograph, so measuring her heart rate. We're measuring galvanic skin response here. She's got EMG recordings on the corrugator muscles, which are frowning, and these are the zygomatic muscles for smiling. And so we gaining a whole lot of information. So we've got heart rate coming here. We've got the galvanic skin response. We've got EMGs from the smile and the eyes. We can calculate heart rate variability. We can take breathing values as well. I can wear a harness and we can measure their breathing rate. I think might be this one. And then the students can add comments in the file when the student is being made angry, or when they are being stressed, or when they're relaxed. And how we try and use this in an engaging way then is to set them up to use it as a lie detector. So the students have to record the response from the two EMG signals, the sympathetic skin response or the galvanic skin response, the heart rate variability, and the respiration rate. They make some measurements under different conditions to see how they change. And then the student is put in a stressful scenario. So they either have to lie about something, so they're given a story and have to tell the truth or lie. Or what actually has proven more efficient is that we tell the students that they've got an unplanned presentation to give in 5 minutes, and we've got one person coming and they're going to be next to do it. And that's always very effective in stressing them out. And we can detect that stress with this lie detection. So all of those things work really well through the Power Lab. I think the benefit for us is that it's very flexible. It's got a whole variety of ways to collect inputs, so like the galvanic skin response. It's a whole bunch of things that I didn't show, but we use these things to do respiration labs, temperature labs, because there are transducers that can collect all of that information and then feed it into the Power Lab. So it makes it quick to build a new lab and to get something up. All of those different transducers mean that you've got a big range of different types of data you can collect. We collect isolated muscles dissected out from different animals as well. So we can measure a wide variety of inputs. From a student point of view, it's pretty robust. So it's stable, it's easy to learn. And once they've dealt with the software once or twice, then they've become very confident in making measurements using the data pad to extract data and using the various add-on analyses like the Fourier transforms to see frequency components. So it's well suited for sort of introductory courses as well. And a big one for us is that it's rated for use with humans, which means that we can actually get the students taking parts in the pracs. They can measure their own responses or that of their colleagues, and that makes it much more fun and immediate. Gets us around the problem of needing animal ethics if we were to do a lot of these experiments with animal tissue. And then the chart reader software, which students can use at home, they can install it themselves, is very valuable. It means that they can take things home, work on it for a prac report. And it meant that during COVID-19, we could continue doing much of what we were doing. We had the electronic lab books. We, instead of the students collecting data and putting it in there, we put pre-existing data in, and then they could analyze it themselves using the chart reader, and then still do all of this, answer the same questions and come to the same conclusions that we would normally expect them to do. So what I'll do now is throw to Mohit, who's going to drill down a little bit more into some of the pracs they do. I've given you a quick overview of some physiology stuff, and now you hear a bit more detail from Mohit, I think.
Yeah, thanks for that, Richard. If you can stop sharing, I can share.
Stop sharing, yeah.
Also, I just realized I had the chat function disabled. So if anyone does have any questions, you can put it in the chat now. I'm so sorry.
Well, good. So hopefully you can see my screen now.
Yep, and your sound's good now.
Oh, great. Perfect. Yeah, look, thanks, Richard. Thanks to Shania and Parimal for this organizing this webinar, and for that introduction you gave for us earlier. So Richard made it very easy for me, and because he's coming perfectly from the physiology end, and you see my title up there. Richard says we work very often, we work very close together, and I think that's the case for physiology and engineering, that they are very close together, and you can learn a lot of physiology through engineering. What we do here in the Graduate School of Biomedical Engineering is try to inculcate some of the physiology principles as well to the engineers, but we try to leave them thinking more about the engineering aspects of what they're doing. So hopefully I'll tell you a bit more about that. But what I'm hoping today to cover is a little bit of an introduction of the field that I come from, which is medical bionics, and then tell you a little bit about the course that I'm coordinating here at UNSW in biomedical engineering, and then particularly drill down into this one particular laboratory that we do with EMG signals, and how we have students use the hardware and software to perform that lab, and then tell you a little bit about what students find and what students feel about their learning and how they reflect on their learning. So, okay, so a little bit about the field. So bionics is essentially a sort of like a portmanteau word, which comes from two different disciplines. It's really about combining biology and electronics. And in my context, it has been all about creating devices. So technology for a range of different applications, technology to perhaps monitor signals from the body, technology to have drug release through an electronic device, and of course the neuroprosthesis area, which is where I come from, which is using electrical stimulation to restore a lost sense, and particularly, like, let's say, for chronic pain or vision or hearing. So bionics is sort of this term. I want to make it clear that many people have, it's a term used quite often in the general public, and there's a different perception perhaps in the general public of what a bionic device is, but really it is, if you integrate biology and electronics, you can call it a bionic device. Probably the most famous example that a lot of our audience would be familiar with is the cochlear implant. So this is a device that's from Cochlear Limited, a company right here in Sydney that manufactures this device, and it uses electrical stimulation of the inner ear. So where you can see this electrode array is inside the inner ear, and there's a implanted stimulator behind the head, just under the skin, and that gets power and data from an external component. So this is the external sound processor that then wirelessly transmits power and data to this internal implant. And what this implant does is that you have electrical current that is passed down this electrode array, and it excites neurons that are within the cochlea, and that gives the perception of hearing to someone who may have lost that perception due to trauma or disease. So I call it the world's most successful, really, implantable bionic device. It's a perfect example of biology and electronics and engineering coming together to, solve a problem in medicine. Perhaps for some of the more mature audience in the field, you might be familiar with this story. So this is course, a show in the 70s called The $6 Million Man. Nothing is available for 6 million these days, though. But back then, it created this whole, you can call interest and hype surrounding bionics. So this particular, in this show, the character gets, all sorts of bionic implants after having an accident. And I think it really sparked some inspiration in the field. So the 70s was all about integrating human and machine. And I think that's 50 years later, we're really seeing some of that happen. Here are some other examples. I told you about the cochlear implant, but now you can pretty much have devices that do all sorts of things. So you can have deep brain stimulation, which treats symptoms of Parkinson's disease. There's already FDA approved devices to treat epilepsy, spinal cord stimulators, even for urinary incontinence, chronic pain, vision, you name it. There's probably not enough space on this slide to actually tell you about all the med tech and all the devices that exist today in this form. So that's about my field. So how does my course relate to my field? So it's actually one of the most lucky things I feel I am in, because I get to teach what I research and breathe day in, day out in my research. And so I teach this in the form of a course called it Bionics and Neuromodulation, of course. And this is a level 9 course, which is an elective. So it's quite an advanced elective. And the way the biomed degree works at UNSW is that it's a 5-year undergrad, postgrad combined degree. So students either take it as a 5-year dual degree, or they can also do a standalone postgraduate degree. So both of these cohorts of students can take this course as an elective. It runs across 9 weeks. The other thing I should mention is the actual engineers who are coming into this course can be from any sort of discipline. So of course, engineering is a very large discipline, and biomedical engineering is in itself very large as well. But we can have we can basically have chemical engineers, bioinformatics, software, electrical, telecommunications, mechanical, mechatronics, and even material science and engineering all coming together to do biomedical engineering. So it's quite a diverse audience. Very few med science students will take this, because I guess Richard's got it covered in med science with the physiology aspects, but we will have a lot of engineering students from all sorts of engineering disciplines taking this course. So that's something we always have to keep in mind, is that although we are teaching a very specialized field of bionics, we are teaching it to a wide audience. These are some of the learning objectives in my course. So I do believe a lot that students should have, by the end of the course, they should have achieved these things. So we even cover a lot of, right from, the knowledge competencies of Engineers Australia. So this course is part of the accredited biomedical engineering degree. So these are the stage 1 competencies that Engineers Australia expects every engineer to have. And the 1-point sort of competencies are more knowledge driven. The 2-point competencies are more application driven, and the 3-point competencies are more communication driven. So you can see the course does cover a wide range of, right from having the theoretical concepts and theoretical knowledge all the way to applying those concepts in the course, and then of course expressing and communicating that very effectively. And so labs is quite a heavy component of this course. It actually makes up 40% of the course, and each of these labs, I only have 2 labs in this course. Each of them run for 3 weeks each, and they make up 40% of the overall course. So it says no final exam in this course. It's more about application and laboratory. And as you can see, this is the key laboratory, one of the key laboratories they have, in which we make use of AD Instruments gear. The way the labs are structured, so Richard showed a photograph with hundreds of students in the class. Ours are a little bit more intimate. We only have about 28 students. So I'll have around 7 to 8 teams of 3 to 4 students, and so I'll have them seated on separate benches. So they're all working in a team, and it's actually quite a diverse team. So as I said earlier, because we have so many diverse backgrounds, we actually intentionally put different engineers together. So we never have 2 electrical engineers in one team or 2 mechanical engineers in one team. We will always have a diverse team. So they are created intentionally. And I think that's where the best stuff happens, is when you actually have diverse disciplines coming together and they all have to solve the problem. Someone will know the electrical side of things, whereas someone else may have, may know about materials, for example. As I said, it's a master's level course with 3 hours of working time. One of the big things we do in the lab is actually get engineers to reflect. It's one of the hardest things for engineers to do, actually. It's when they sit down and actually think about what they've done and they talk about it. And usually it's very hard to get engineers to come out of their comfort zone because they don't like talking about it. They usually just like working by themselves. They can put a report at the end, but this thing actually forces them to come out of their comfort zone and participate in discussion sessions at the end of the lab. So they actually tell us what they've learned. The labs are on about 15 student staff ratio. So there's two staff members, including me, in the lab. And as I said, because it's weighted 40%, we get them to do very extensive lab reports in this course. Okay, so a bit about how this lab runs, this particular EMG lab, and how we do, how we get them to use the 80 Instruments gear. So we do provide them with an instructional guide. It has stuff written in it, but in terms of the setup of the experiment, it has it has very little in it. So because these are engineers and because they're sort of year level 3 and above, we do expect them to have some fundamental engineering knowledge. So we tell them, about the physiology, and we tell them about what EMG signals are, how EMG signals apply to the field of bionics. For example, the best application is in myoelectric prostheses, which are, artificial arms that use EMG signals to move the artificial arm, for example. We tell them about different ways of recording activity, how to actually place electrodes for recording from the biceps, which is what they need to do. Give them a description of the 3 experiments that they will do with EMG. That's why it goes for 3 weeks, because we actually have quite a few experiments. Tell them about some basic analysis and give them reading, but they're really expected to then dive in once they start collecting their data and they analyze their data. So 3 experiments we do is, one is for reaction time. So we either connect the gear to an LED flash or a sound device, and we get them to, flex their biceps in response to a stimulus. And either the flash or the sound on its own, or the flash and sound together as a multisensory stimulus. And they actually then calculate reaction times and see how the brain integrates 2 different senses together. They also do an experiment where they study the force EMG relationship. So we tell them you're going to have a gym workout because we actually give them weights, and we tell them to do weight workouts while recording EMG. And we look at those muscles or the electrical activity of those muscles during this application of this force. And the last one is really the most fun for students from an engineering standpoint, because we actually use EMG in a closed loop system. And I'll talk about it a little bit later on how we use the EMG signals as feedback to control an electrical stimulus. It's one of the most fun parts of the lab. So in the lab, we make use of the ultimate teaching system, and not all the components of the teaching system are used, but those are the different components we're using in this particular laboratory. So it's, as Richard mentioned, it's really very, very versatile, this particular piece of hardware, because it has so many range of inputs and outputs. And yeah, I think its biggest plus point is that it's human grade. So even for electrical stimulation, that it has an isolated stimulator, and that really opens up a lot to what we can really do with this setup. So this is kind of the hardware setup. This is the typical electrode placement we would have, and this is a typical recording. So here you see a trigger that's been sent out to produce a sound. They hear the sound, and in response to the sound, they basically flex their biceps, and they do that multiple number of times repeatedly. So as I said earlier, these are engineers, so we expect them to configure their own settings. So we don't tell them a lot about settings. We don't tell them about, noise. They're meant to basically look at the quality of the signal and choose the right filter settings so that they are able to record high quality signals. They're supposed to write their code. This is a very simple code that just says randomize the repeats every time. So we get them to write this code. They, as I said, they choose their own filters. They're meant to know what these filter settings are. Of course, we do help them, but that's the part of the engineering that we're sort of saying, well, if you want to record a biological, physiological signal, you have to know the electrical conditions behind it. So that's how we sort of teach those electrical concepts. And this is what we would typically get. So students do record like raw signals, but then they also record filtered signals to make it cleaner. And then they go and analyze these filtered signals later to come up with a whole sorts of things. This is sort of the diagram of the setup we have for the force EMG experiment. So as I said, we are recording EMG, but we're placing weights on their arms. And here you can see a typical recording where each time a weight is placed, there's an indicator, and then that you can actually see the EMG signal grow in amplitude as well as in frequency. And we get them to analyze that later on. And then the third experiment, as I said, is the most fun. So what we do here is we connect up the EMG to one student, and we connect up the electrical stimulator to another student, and we attach the stimulating electrode to their ulnar nerves. We establish a little threshold of current that we put out through this to make their fingers twitch. And once we have established that threshold, we ask the students to apply a automated analysis of the EMG in real time to then trigger the stimulator. So it's quite fun when it works. What happens is the two students face each other. They have no clue what's happening. And then when one student flexes their biceps, the other student's arms twitch. So it's essentially a complete closed loop system where there's a recording happening from one person, but then that triggers and moves another person's arm. So students, when they see that, it's very fun and engaging, but B, I think they very much then get in their heads the concept of having physiological feedback to trigger some activity or trigger some stimulus. And that concept gets drilled in their heads very nicely. Okay, and all the data is then exported. They export by themselves to MATLAB, and they're meant to write MATLAB code for doing quite some sophisticated analysis. So they look at rectification, filtering, RMS calculations. Here's a plot of how RMS amplitude changes with the force that they apply. And this is also looking at frequency domain analysis to look at muscle fatigue. So they look at mean and median frequency plots to see how the muscle is fatiguing over time as more weights are added. So quite a lot of analysis and signal processing that they have to do. And at the end of it, to get their marks, they have to write quite an extensive report. So this is just to show you. Turn the laser off. Yeah. Okay, it should scroll. So you can see here it's scrolling. So we get them to write a full-on report that includes introduction to the field, how they performed their experiment. So each team may perform it slightly differently. They're supposed to tell us the exact methods on how they perform the experiment. And then they present us the results of the experiment, just like how they would write a journal article almost. And finally, they have to write a discussion and a conclusion section, but also then a big reflection section that carries a lot of marks. And this is where the students really have to reflect on their learning and tell us what they learned, what aspects of the lab was engaging to them, and how it links what they've learned in engineering and physiology, how it is all brought together. So from an academic perspective, the learning hopefully is that students get to reinforce the concepts and devices that they introduce in lectures. So as I mentioned earlier, one of the devices that they lectured about is this myoelectric prosthesis that uses similar electrodes placed on muscles and records EMG activity to control an electric hand. So that's what they learn about in the lecture. And then hopefully in the lab, what they're then seeing is that reinforcement of that concept that you can use a physiological signal like EMG to control something. So these are really the things that from an academic perspective, we think that students will learn this. But when we look from a student perspective, what they learn, it's very intriguing and very interesting to read about. So this is a part of a reflection from a student who did this lab. And that particular student said that, when they took physiology the year before, they did something with a frog's leg and watched it twitch. And it really piqued their interest. And then it continued in anatomy, which is also a course that runs in Richard School. And they were able to, view all these things. And then now, having done this engineering laboratory, they've really combined their previous experience and brought it into context to how it relates to engineering. So I think this is a very nice example of students actually, not only engaging with the lab, but actually learning from and bringing it together from their previous knowledge through other courses, which is really nice to see. This is another one where this student has reflected all the way to the first year. So this student is taking this course in fourth year, but they say that I applied previous knowledge to my current task. I gained new knowledge that could have been applied to an old task, and I now see how the bionic hand that I made in Eng 1000, how that could be actually improved. So this student experienced trying to make a bionic hand in first year would have really failed, because but then after doing the EMG laboratory, they were able to get that knowledge that they could now go and apply it back. So it's really nice to see how they're reflecting all the way to their first year. This is some of the feedback we get. So it reaffirms the course learning objectives, because the students say, I really enjoyed writing the lab reports. It required me to do research, and so it almost forced them to understand and link, the physiology and the engineering concepts together, and also improve their report writing skills. Another student said, this actually wrote in their feedback that they understood the diversity in biomedical engineering, and so this student said it was not from an electrical background, and the subject was going to be hard, but the real life applicability and the interesting topics, those are the things that made it doable. So it's challenging, but also it's engaging, and it forces them to come out of their comfort zone. And then lastly, this is another reflection of another student where they're reflecting through practical experience. So they're saying the labs are structured that allow students to understand and apply course content. And of course, because there's very little exam, there's no exam virtually. What they would normally do is they cram up at the end and for an exam and then forget everything the minute the exam's over. But in this particular case, because they've done it through a practical means, they retain the information much longer. And that is the hope, of course, that we have as well. So that's what occurs in my course, but we don't only use 80 instruments in my course. This is just a quick example of another course that makes use of the same teaching system. It's called Biosensors and Transducers. So here they actually try to teach concepts of how does spirometry work, what is it used for, how do you calibrate, and this is sort of the waveforms that they get them to record. So they use the spirometer module that comes with the teaching kit, and they get students to blow into it, and so you get this expiration curve, and they ask students to go and analyze that. Okay, so in summary, medical bionics, very exciting and exploding field for the last 40-50 years. And certainly like to shout out that 80 instruments have played a very big role in enabling our teaching to be quite effective, because it's only with this sort of gear that makes it simple, makes it seamless data acquisition, that we can have students doing these labs. And as hopefully you saw with the examples, that the learning, engagement, and feedback from students is really positive from this. So with that, thanks for attending. Thanks for all your attention. And I'm pretty sure Richard and I would be more than happy to answer any questions.
Perfect. Thank you so much, Richard and Mohit. What I'll do is I'll share the slide with both of your details on there now. So just want to, since we're waiting on people to ask questions, I'll actually ask you guys both a question as well. How did you guys find it using LabChart and Power Labs to customize your own labs?
Richard, you want to go first? Yeah.
Sure, yeah. So I guess that's sort of what I was alluding to, that because there's a pretty robust software package and there's a variety of different transduction options, it's, most of those labs I presented, we developed ourselves, and they kind of evolve from year to year, which I think is really one of the big strengths, just that students can relate to a pretty standardized box and software package, but then do things ranging from measuring lung function to recording from single neurons to making other people twitch when someone is moving their arm. So yeah, no, it facilitates all that kind of development work, I think.
Yeah, I'd kind of second that. I think the biggest advantage of the gear is its flexibility, and that it's open-endedness. So it's not locked into any particular thing. And so the imagination is really the key here. You can almost design whatever kind of prac you want to design, really, because you've got the range of outputs and inputs available to you. And so that's I think is it's the power. The other side of the thing is the intuitiveness of the lab chart. So students, I'm pretty sure even that this is the case with physiology students, that it wouldn't take long for them to fire up a Power Lab and get into it and get going. So it's not like they need 5 hours that it's so specialized that you need 5 hours to set it up. You can get going very quickly. So I think those two things, that's why I said it makes it really seamless in terms of data acquisition.
And the ability to get your data out to then do further analysis and other packages, where some things try and hold you inside their ecosystem. I mean, that's a big strength, I think, too.
Correct.
Perfect. Just checking the chat. No one's asked anything. Could probably give it a few minutes more.
They may be contacting you to buy Power Labs.
Yeah, so what we can do is it's almost 3, but everyone has your email addresses on here. So if anyone does need to even get in contact with Mohit or Richard, you guys can always get into contact with them.
We just see there's just one question coming now.
Oh, perfect.
Just how much support time students need to learn the setups. Yeah, so thanks Manish for that question. So yeah, it's do they learn on the fly during the lab? Yes, the answer is yes. So we give them, at least in engineering, we give them that lab instruction beforehand, but they've never seen the Power Lab ever. And they've never seen lab chart ever. So the first thing they come in, they get going in about 20 minutes. Literally, they get going from 0 to recording data in 20 minutes. That's how powerful it is. And because again, they're working in teams, there will always be someone, who'll say, oh, I know how to stick the electrodes on. I've done that before. And someone else will say, well, I know what a BNC connector is. So I've used that before. So I can use that. And I know what a function generator is, and I can use that. So it's really that multidisciplinary team approach makes the whole team learn really fast. So it's during the lab itself, they learn on the fly. And then when they have finished that lab, they go back and reflect on what they have just done. And that improves the learning even more, because they go back to what they've done and they read about it in the literature, and then they have these really nice aha moments where they said, oh, I saw a news article, I read a news article or saw a news story about this particular thing, and now I've done this in the lab and I can now link these together. So it's really nice when that does happen. I'm assuming physiology students, Richard. Yeah.
Very quick one on behalf of other attendees. Mohit and Richard both, are you open to share your protocols or experimental setup protocols with others if required, or if they need any sort of support and stuff like that?
Yeah, absolutely.
Absolutely.
Yeah,
I think that's our motivation for coming on today, really, is that, education should be collaborative. We're all trying to, help enrich the world and produce great students. And I think that means we should be sharing our protocols and methodology. So yeah, happy to share settings files or prac notes or whatever would be useful.
Yep.
Thanks, Richard and Mohit, for your kind gesture here. So any attendees who wish to contact Mohit or Richard can contact them directly, or alternatively, they can contact your ADI rep and we can put you in touch with both of them as well. Thank you.